Good evening, students. So, in this class, we are going to discuss with one more most important chapter in botany. So, that is photosynthesis in plants. We will get around 3 marks from this chapter. Okay, we will get around 3 marks from this chapter. Okay, 3 to 4 marks in CET. Fine. So, this is the most important chapter for board as well as CET and NEET. So photosynthesis in higher plants. So regarding photosynthesis in higher plants, we started studying regarding photosynthesis from 7th standard. Right. So what do you mean by photosynthesis? It is nothing but the preparation of food from the plants in presence of sunlight with the help of what we can say, with the reaction of carbon dioxide and water in the chloroplast. Okay, so that is called as what? Photosynthesis, what we call. Okay, so here photosynthesis in higher plants, what majorly we have to concentrate in this lesson. So let me brush up some concepts first, then later we shall move on to the questions. Okay, so as you can see here, okay. So first we are going to study an organelle where photosynthesis takes place. Which is that organelle? It is nothing but a chloroplast, right? So the most active photosynthetic tissue in higher plant is mesophyll of leaves. So you saw, I think you are familiar with the monocot as well as dicot leaf. Okay. So in the anatomy of flowering plants, we studied regarding it. So the tissue present in the leaf, it is called as what? Mesophyll tissue. The leaf tissue is called mesophyll tissue. Okay. So the most active photosynthetic tissue is a mesophyll tissue of the leaves. So what happens here? Mesophyll cells. So the cells which are present in the mesophyll tissues, alva. So mesophyll cells will be having many chloroplasts compared to other cells. So chloroplast will be present in all the green parts of the plant. There may be over 500,000 of chloroplasts. See here, how many? 500,000 of chloroplast per square millimeter of leaf surface. So here you can see in the higher plants, chlorophyte will be in a discoid or lens shape. You can see the lens shape, alva. So they are usually 4 to 10 millimeter in diameter, 1 to 3 millimeter in thickness. So when you see the diagram, you will understand, my dear boys and girls. So here you can see it is a membranous structure. So here we can see the outer membrane, right? And here you can see the inner membrane. Howda? So between outer and inner membrane, a space is present which is called intermembrane space. Fine. So inside the over, after the inner membrane, it has a fluid which is called as stroma. It will be aqueous in nature. It is a fluidic in nature. Howda? Then you can see oval shaped structures here, right? So each oval shaped structure, okay, this is called thylakoid. Then the group of thylakoids are arranged one above the other like a pile of a coin. ಅಂದ್ರೆ ಒಂದು ಕಾಯಿನ್ ಆದ್ಮೇಲೆ ಇನ್ನೊಂದು ಕಾಯಿನ್, ಅದ್ರ ಮೇಲೆ ನಾವು ಹೇಗೆ ಇಡ್ತೀವೋ ಯಾವ ರೀತಿ ಆ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಣಿಸುತ್ತೆ, ಕಾಯಿನ್ ಮೇಲೆ ಕಾಯಿನ್ ಇಟ್ಟಾಗ ಅದೇ ರೀತಿ ಈ ಥೈಲಕಾಯ್ಡ್ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಣಿಸುತ್ತೆ. ವೆನ್ ದಿಸ್ ಈಸ್ ಅ ಗ್ರೂಪ್ ಆಫ್ ಅ ಥೈಲಕಾಯ್ಡ್ ವಿಚ್ ಆರ್ ಅರೇಂಜ್ಡ್ ಒನ್ ಅಬೌವ್ ದ ಅದರ್, ಇದಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೇವೆ? ಗ್ರಾನಮ್ ಅಂತ ಕರಿತೇವೆ, right? So this grana, so various grana will be there na. These grana will be interconnected by a lamella, by a structure. This structure is called as lamella anta kariteve. So inside the thylakoid, a lumen is present. Okay, space is present which is called lumen, where photosynthesis takes place. So ಈ ಸ್ಟ್ರೋಮಾದಲ್ಲಿ ಒಂದು ರೀತಿಯ ಫೋಟೋಸಿಂಥಸಿಸ್ ಆದ್ರೆ ಥೈಲಕಾಯ್ಡ್ ಅಲ್ಲಿ ಇನ್ನೊಂದು ರೀತಿಯ ಫೋಟೋಸಿಂಥಸಿಸ್ ಆಗತ್ತೆ. ಓಕೆನಾ? So which kind of photosynthesis takes place in thylakoid and stroma? We are going to learn now. All right. So see here. Chloroplast pigments anta baratte. Let me take this type so that you will understand. Yeah. So what is the main thing of chloroplast? The main thing of chloroplast is giving green color. Why it gives green color or any color? It is due to the presence of the chemical substances which are called, we can call it as pigments. ಅದಕ್ಕೆ ನಾವೆಂತ ಕರಿತೇವೆ, ಪಿಗ್ಮೆಂಟ್ಸ್ ಅಂತ ಕರಿತೇವೆ. ಓಕೆನಾ? ಸೊ ಹಿಯರ್ ಯು ಕ್ಯಾನ್ ಸಿ ಪಿಗ್ಮೆಂಟ್ಸ್ ಅಂದ್ರೆ ಏನು? These are organic molecules. What kind of molecules these are? Organic molecules. Okay, these are organic molecules. What is the function of these pigments? Their function is they absorb light of a specific wavelength, we can say. They absorb light of a specific wavelength. Just a minute. Yeah. Right. Then after that, ಯಾವ ರೀತಿ ಒಂದು ವೇವ್ಲೆಂತ್ in the visible region, so that light should be in a visible region, right? The chloroplast pigments are fat soluble. They are soluble in fat and they are located in the lipid part of thylakoid membranes. So you saw thylakoid, right? So in the thylakoid will be covered by a membrane. In the lipid part of a thylakoid membrane, these pigments are present. So how many pigments we have? So here you can see chlorophyll A, chlorophyll B, chlorophyll C, right? Chlorophyll D, chlorophyll E. So these are the chemical formulas. Fine. So it varies in the number of what we can say oxygen, hydrogen and oxygen. Then yeah, hydrogen and oxygen vary ಆಗಿರೋದ್ರಿಂದ we have different kind of pigments. So distribution anta ನೋಡಿದಾಗ all photosynthetic organisms except photosynthetic bacteria. Chlorophyll A is present in all photosynthetic organism. Either it will be algae, bacteria or any plant. Then chlorophyll B anta bandre chlorophyta, ಯುಗ್ಲಿನೋಫೈಟಾ ಮತ್ತೆ ಎಲ್ಲ ಹೈಯರ್ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲೂ ನಮಗೆ ಕ್ಲೋರೋಫಿಲ್ ಬಿ ಕಾಣಿಸ್ತಿಗತ್ತೆ. ದೆನ್ ಕ್ಲೋರೋಫಿಲ್ ಸಿ ಅಂತ ಬಂದ್ರೆ ಇಟ್ ಈಸ್ ಫೌಂಡ್ ಇನ್ ಬ್ರೌನ್ ಅಲಿಗೆ ವಿಚ್ ಗಿವ್ಸ್ ಬ್ರೌನ್ ಕಲರ್, ಡೈಅಟಮ್ಸ್ ಅಂಡ್ ಪೈರೋಫೈಟಾ. ಕ್ಲೋರೋಫಿಲ್ ಡಿ ಅಂದ್ರೆ ಇಟ್ ಈಸ್ ಫೌಂಡ್ ಇನ್ ರೆಡ್ ಅಲಿಗೆ, ರೋಡೋಫೈಟಾ, ರೋಡೋಫಿಲ್ ಅಂತಾನು ಕರಿತೀವಿ. ಕ್ಲೋರೋಫಿಲ್ ಇ ಅಂತ ಬಂದಾಗ ಕ್ಸಾಂಥೋಫಿಲ್, ಸೊ ಇದು ಕ್ಸಾಂಥೋಫೈಟಾದಲ್ಲಿ ಕಾಣಿಸಿಗತ್ತೆ. So these are the types of what we can say the pigments. Okay, now next. Again we have, you can see bacteriochlorophyll anta baratte, which is present in purple photosynthetic factor. Please note down, very important, bacteriochlorophyll. Chlorobium chlorophyll anta baratte, which is also called as bacterioviridin, which is present in green sulfur bacteria. Fine. Then okay, regarding this we will discuss later. Now we shall discuss the questions. Okay, carbon 4 and carbon C3 and C4 plants. Okay, fine. Let us proceed with the first question of today. Very easy question. The process in which green plants synthesize organic food by utilizing carbon dioxide and water as raw materials, okay, in the presence of sunlight, what it is called as? Anyone? Yes, YouTube students, yeah, come on, what it is called as? Very good, it is nothing but a photosynthesis. Option C is a right answer. Next question, which is correct regarding photosynthesis? Few statements are given here, so which is correct regarding photosynthesis? We should say carbon dioxide is obtained, I'm sorry, yeah, carbon dioxide is obtained from the atmosphere. B, water is absorbed from the soil through the stem system. C, sunlight is trapped by pigments called xanthophyll. D, chlorophyll absorbs green light. So we have to find which is the correct statement regarding photosynthesis. See, carbon dioxide is obtained from the raw atmosphere. Oh, sorry, from the atmosphere, right? So here carbon dioxide means what? It is a CO2, okay, which is a what we can say reactant in the photosynthesis or a raw material in the photosynthesis. Alva. Next, water is absorbed from the soil through the stem system. Sunlight is trapped by pigments called xanthophyll. So usually the sunlight is trapped by the pigments called chlorophyll irutte. Chlorophyll absorbs green light anta. So which light is absorbed, that light will not be reflected, right? So here when you can see the plants, majority of the plants are green in color. So obviously chlorophyll will not absorb the green light, right? So let us check the answer for this question. So right answer for this question is carbon dioxide is obtained from the atmosphere. Water is absorbed from the soil through the stem system. No. So usually the water is absorbed from the root system, right? Then sunlight is trapped by pigments called xanthophyll. So usually a pigments called chlorophyll will absorb the sunlight. All right. So here this already I explained. It absorbs the red light and it reflects the green light. Fine. So option A is a right answer in this question. Let us proceed to the next question here. Okay. So dark reactions of photosynthesis are actually not totally independent of light as. So in the photosynthesis, we have two reactions. One is dark reaction. Fine. The one is light reaction. Okay. So dark reaction of photosynthesis are actually not totally independent of light as. Yes, YouTube students, you can answer in the chat box. So here, the initial reactions occur in the presence of light. The reaction will be inhibited by the presence of light. They can occur during the day as well. They utilize the products of the light reaction. ಸೊ ಡಾರ್ಕ್ ರಿಯಾಕ್ಷನ್ ಅಂತ ಹೆಸರಿದ್ರೆ ಸೊ ಯೂಶ್ವಲಿ ಸನ್ಲೈಟ್ ಅನ್ನ ಉಪಯೋಗಿಸೋದಿಲ್ಲ ಅಂತ ತಲೆಯಲ್ಲಿ ಬರುತ್ತೆ. ಆದ್ರೂ ಕೂಡ ಡಾರ್ಕ್ ರಿಯಾಕ್ಷನ್ ಇಸ್ ನಾಟ್ ಟೋಟಲಿ ಇಂಡಿಪೆಂಡೆಂಟ್ ಆಫ್ ಲೈಟ್ ಅಂತ. ಯಾಕೆ? ರೀಡ್ ಔಟ್ ದ ಆಪ್ಷನ್ಸ್ ಹಿಯರ್. Read out all the options and let me know the answer. Yeah, I'm waiting. So this is third chapter in botany, right? So already it will be completed at your respective colleges. So you will be having the theoretical knowledge of this chapter. So you can answer this question. Yes, come on. Dark reactions of photosynthesis are actually not totally independent of light as. Yes. Okay, Anuradha answered it as option B. The reaction will be inhibited by the presence of light. Okay, fine. So let us check the answer here. The right answer is option D. They utilize the products of light reaction. So whatever products will be released or will be formed during the light reaction, those products are only, are necessary for the dark reaction to happen. It is not that the reaction will be inhibited by the presence of light. They asked dark reactions even though it is not completely independent of light. So the right answer is here. They utilize the products of light reaction. Fine, just a minute. So let me explain you the dark reaction and the light reaction. I will just brush up the concept. Who don't know it, you can just go through it. Okay. So here you can see the mechanism of the photosynthesis, right? So when we study regarding the mechanism of the photosynthesis, here we have two processes in it. One is light reaction, or which is called photochemical phase. The other is dark reaction, or it is called biosynthetic phase. Photochemical phase and biosynthetic phase, right? So here photochemical phase andre ಏನು? Just a minute. Hmm. So photochemical phase andre or light reaction andre ಏನು? First, here what happens is absorption of light takes place. Absorption of light ಆದ್ಮೇಲೆ splitting of water, release of oxygen, finally formation of high energy chemical intermediates which are called ATP and NADPH. So these are only used in the dark reaction. So these photosynthetic pigments are organized into two discrete photochemical light harvesting complexes. So ಇಲ್ಲಿ ಲೈಟ್ ಎನರ್ಜಿಯನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ಕೋಬೇಕು ಅಲ್ವಾ, ಯಾರು ಈ ಪಿಗ್ಮೆಂಟ್ಸ್ ಗಳು, ಹೌದಾ? ಸೊ ಡ್ಯೂರಿಂಗ್ ದ ಕೋರ್ಸ್ ಆಫ್ ಲೈಟ್ ರಿಯಾಕ್ಷನ್ ಈ ಪಿಗ್ಮೆಂಟ್ಸ್ ಗಳು ಏನ್ ಮಾಡ್ತವೆ, ದೇ ವಿಲ್ ಅಬ್ಸಾರ್ಬ್ ದ light in the quantazones of the granathylakoids. Granathylakoids ಅಲ್ಲಿ quantazones ಮುಖಾಂತರ they absorb the what light, or the light is trapped. ಈ photosynthetic pigments, these are organized into discrete photochemical light harvesting complexes. ಸೊ ಇದಕ್ಕೆ ನಾವು photosystem 1 ಮತ್ತೆ photosystem 2 ಅಂತ ಕರಿತೇವೆ, ಹೌದಾ? ಸೊ ಇಲ್ಲಿ the light harvesting complexes, or what we can say photosystems, these are made up of 100 of pigment molecules. ಒಂದು photosystem 100 of pigment molecules ಇಂದ ಮಾಡಲ್ಪಟ್ಟಿರುತ್ತೆ and bonded by proteins. Each photosystem has photocenter or reaction center where actually reaction takes place. Fine. So let us continue. So ಇಲ್ಲಿ ನೋಡಿ, these photosynthesis are named according to the sequence of their discovery, not in the sequence of the light reaction. So here we can see pigment molecules are there. So these pigment molecules, what they are doing, they are absorbing the sunlight in the form of a photons, right? So when these pigment molecule, these are getting excited, and here the energy or the thing will be accepted by the primary acceptor. So here you can read here, the reaction center is different in both photosystem. Photosystem 1 ಎಲ್ಲಾಗತ್ತೆ, or a chlorophyll A ಜಾಸ್ತಿ ಇರತ್ತೆ ಎಲ್ಲಿ, photosystem 1 ಅಲ್ಲಿ. ಸೊ ಅಲ್ಲಿ ಯಾವ ರೀತಿಯ ಲೈಟ್ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಆಗತ್ತೆ, 700 ನ್ಯಾನೋಮೀಟರ್. Then PS2 photosystem 2 ಅಲ್ಲಿ the reaction center has absorption peak 680 ನ್ಯಾನೋಮೀಟರ್ ಇರುತ್ತೆ. ಇದಾದ್ಮೇಲೆ ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಆಗತ್ತೆ. The photosynthetic electron transport chain initiate ಯಾವಾಗಿಂದ ಸ್ಟಾರ್ಟ್ ಆಗತ್ತೆ, when it absorbs the light by photosystem 2. The red light of wavelength 680 nanometer is absorbed by the reaction by PS2, due to which electrons becomes excited. So ಯಾವಾಗ ಲೈಟ್ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಆಗತ್ತೋ, ಎನರ್ಜಿ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಆಗತ್ತೋ, ಅಲ್ಲಿ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಗಳು ಎನರ್ಜಿಯನ್ನ ಪಡ್ಕೊಂಡು ಏನ್ ಮಾಡ್ತವೆ, ಎಕ್ಸೈಟ್ ಆಗ್ತವೆ. ದೇ ವಿಲ್ ಬಿಕಮ್ ಮೋರ್ ಆಕ್ಟಿವ್, ದೇ ವಿಲ್ ಗೆಟ್ ಎಕ್ಸೈಟೆಡ್. ಎಕ್ಸೈಟ್ ಆದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ, ಅವು ಅದೇ ಶೆಲ್ ಅಲ್ಲಿ ಇರೋದಿಲ್ಲ. ಇನ್ ಕೆಮಿಸ್ಟ್ರಿ ವಿ ಲರ್ನ್ಟ್ ಅಲ್ವಾ, electrons will be revolving in the shells. So when they are excited, they will shift to they will jump into the other orbit away from the atomic nucleus. Right? ಸ್ವಲ್ಪ ದೂರ ಹೋಗ್ತವೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಂದ, ಯಾಕೆಂದ್ರೆ ಅವು ಎಕ್ಸೈಟೆಡ್ ಸ್ಟೇಟ್ ಅಲ್ಲಿ ಇವೆ. ಸೊ ಯಾವ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಎಕ್ಸೈಟ್ ಆಗಿದೆನೋ, these electrons are then picked up by an electron transporter. ಅಲ್ಲಿ ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ಇರುತ್ತೆ which accepts or the electrons, excited electrons. ಅದಕ್ಕೆ ನಾವು ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟರ್ ಅಂತ ಕರಿತೇವೆ, ಹೌದಾ? ಓಕೆ, ಎಲೆಕ್ಟ್ರಾನ್ ಎಕ್ಸೆಪ್ಟಾರ್ ಅಂತ ಕರಿತೇವೆ, ವಿಚ್ ಪಾಸಸ್ ದೆಮ್ ಟು ಫರ್ದರ್ ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಸಿಸ್ಟಮ್. ಸೊ ಈ ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಎಕ್ಸೆಪ್ಟಾರ್ ಏನ್ ಮಾಡತ್ತೆ, ಆ ಎಲೆಕ್ಟ್ರಾನ್ ಅನ್ನ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಸಿಸ್ಟಮ್ಗೆ ಪಾಸ್ ಮಾಡತ್ತೆ. ಇಟ್ ಈಸ್ ಟು ಬಿ ನೋಟೆಡ್ ದಟ್ ದಿಸ್ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ಎಲೆಕ್ಟ್ರಾನ್ ಈಸ್ ಡೌನ್ ಹಿಲ್ ಅಕಾರ್ಡಿಂಗ್ ಟು ರೆಡಾಕ್ಸ್ ಪೊಟೆನ್ಶಿಯಲ್ ಸ್ಕೇಲ್. ಸೊ ಇದು ಈ ರೀತಿ, ಇಲ್ಲಿ ನೋಡಿ, ಕ್ಲೋರೋ ಎಲೆಕ್ಟ್ರಾನ್ ಎಕ್ಸೆಪ್ಟರ್ ಎಕ್ಸೆಪ್ಟ್ ಮಾಡ್ಕೊಳತ್ತೆ. ಸೊ ಈ ರೀತಿ ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಸಿಸ್ಟಮ್ ಇಂದ ಅಲ್ಲಿ ಲೈಟ್ ಹಾರ್ವೆಸ್ಟಿಂಗ್ ಸಿಸ್ಟಮ್ ಮತ್ತೆ ಎಲ್ಲಿ ಬರುತ್ತೆ, ಫೋಟೋ ಸಿಸ್ಟಮಿಗೆ ಒನ್ ಬರುತ್ತೆ. ಹೌದಾ? ಅಗೇನ್ ದಿಸ್ ದ ಲೈಟ್ ಹಾರ್ವೆಸ್ಟಿಂಗ್ ಸಿಸ್ಟಮ್ ದಟ್ ಇಸ್ 700 ನ್ಯಾನೋಮೀಟರ್ ಲೈಟ್ ಅಬ್ಸಾರ್ಬ್ ಮಾಡಿಕೊಂಡು ದಿಸ್ ಎಲೆಕ್ಟ್ರಾನ್ ಎಕ್ಸೆಪ್ಟಾರ್ ವಿಲ್ ಡೂ ವಾಟ್, NADP ಪ್ಲಸ್ ಇದ್ದಿದ್ದು ಇಲ್ಲೇನಾಗತ್ತೆ, NADPH ಆಗತ್ತೆ. ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ಇಟ್ ಇಸ್ ಟು ಬಿ ದ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಆಫ್ ದ ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಚೈನ್ ಆರ್ ನಾಟ್ ಯೂಸ್ಡ್ ಅಪ್ ಇನ್ ದ ಚೈನ್, ಇನ್ಸ್ಟೆಡ್ ದೇ ಆರ್ ಫರ್ದರ್ ಪಾಸ್ಟ್. ಸೊ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಗಳು ಇಲ್ಲೇನಾಗ್ತವೆ, ಪಾಸ್ ಆಗ್ತಾ ಹೋಗ್ತವೆ ಒಂದು ಸಿಸ್ಟಮ್ ಇಂದ ಇನ್ನೊಂದು ಸಿಸ್ಟಮ್ಗೆ, ಯೂಸ್ ಆಗೋದಿಲ್ಲ. ದೆನ್ ಸೊ ಇಲ್ಲಿ PS2, the electrons in the reaction center of PS1 also gets excited on getting the red light of wavelength 700 nanometer and get transferred to the electron acceptor with hitter redox potential ಅಂತ. ಸೊ ಇದಾದ್ಮೇಲೆ ಸ್ಪ್ಲಿಟ್ಟಿಂಗ್ ಆಫ್ ವಾಟರ್ ಆಗತ್ತೆ, ವಿಚ್ ವಿ ವಿಲ್ ಕಾಲ್ ಇಟ್ ಆಸ್ ಹೈಡ್ರೋಲೈಸಿಸ್. ಸೊ ದ ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಆರ್ ಕಂಟಿನ್ಯೂಸ್ಲಿ ಸಪ್ಲೈಡ್ ಟು ದ ಫೋಟೋ ಸಿಸ್ಟಮ್ ಟು by available electrons which gets displaced due to splitting of water. Fine. See, this is the reaction of splitting of water. Here you can see. ಇನ್ನೊಂದು photophosphorylation ಅಂತ ಬರುತ್ತೆ. It is the process through which ATP is synthesized from ADP. ADP ಇಂದ ATP ಫಾರ್ಮ್ ಆಗತ್ತೆ. ADP ಮತ್ತೆ ಇನ್ಆರ್ಗ್ಯಾನಿಕ್ IP ಇಂದ. ಹೌದಾ? ಇನ್ಆರ್ಗ್ಯಾನಿಕ್ ಫಾಸ್ಫೇಟ್ ಅಂತ ಕರಿತೇವೆ. ಸೊ ಬೈ ದ ಸೆಲ್ ಆರ್ಗನಲ್ಸ್, ಯಾವ್ದ್ರಿಂದ, ಮೈಟೋಕಾಂಡ್ರಿಯಾ ಮತ್ತೆ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಇಂದ, ವಿತ್ ದ ಹೆಲ್ಪ್ ಆಫ್ ದ ಎನರ್ಜಿ ಫ್ರಮ್ ಸೋಲಾರ್ ರೇಡಿಯೇಷನ್. ಸೊ ಅದಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೇವೆ, ಫೋಟೋಫಾಸ್ಫೋರೈಲೇಷನ್. ATP ಸಿಂಥೆಸಿಸ್ಗೆ ಫೋಟೋಫಾಸ್ಫೋರೈಲೇಷನ್ ಅಂತ ಕರಿತೇವೆ. ಸೊ ಯಾವಾಗ ಫೋಟೋಫಾಸ್ಫೋರೈಲೇಷನ್ ಮೈಟೋಕಾಂಡ್ರಿಯಾದಲ್ಲಾಗತ್ತೆ, ಅದಕ್ಕೆ ಲೈಟಿನ ಅವಶ್ಯಕತೆ ಇರೋದಿಲ್ಲ. ಬಟ್ ಇಟ್ ಯೂಸಸ್ ದ ಎನರ್ಜಿ ಬೈ ಆಕ್ಸಿಡೇಷನ್ ಆಫ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ATP. ಸೊ ಅದೇನ್ ಮಾಡತ್ತೆ, ಅದರಲ್ಲಿರುವ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಅನ್ನ ಆಕ್ಸಿಡೈಸ್ ಮಾಡಿ ಅದರ ಮುಖಾಂತರ ಎನರ್ಜಿಯನ್ನ ಪಡೆದು ATP ಯನ್ನ ಫಾರ್ಮ್ ಮಾಡುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೇವೆ, ಆಕ್ಸಿಡೇಟಿವ್ ಫಾಸ್ಫೋರೈಲೇಷನ್. ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ವೇರ್ ಡಸ್ ಆಕ್ಸಿಡೇಟಿವ್ ಫಾಸ್ಫೋರೈಲೇಷನ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್? It takes place in the mitochondria. Fine. So let us proceed with the questions now, and again we shall come back to the concepts. Fine. So here, idaitalva. Next question. All the following regarding the role of xanthophylls and carotenoids in photosynthesis are correct except. So xanthophylls ಮತ್ತೆ carotenoids ಅನ್ನೋ pigments ಇದೆ. So all of the following regarding the role of xanthophylls or carotenoids is in photosynthesis. So ಕೆಲವೊಂದಿಷ್ಟು ಇಲ್ಲಿ ಅವುಗಳ ಫಂಕ್ಷನ್ ಅನ್ನ ಹೇಳ್ತಾರೆ, except which is a wrong statement regarding xanthophylls and carotenoids ಅಂತ ಕೇಳ್ತಿದ್ದಾರೆ. Option 1, they act as accessory pigments. Option 2, their peak absorption is in blue and red regions of Vibgyor. Option 3, they enable a wider range of wavelength of incoming light to be utilized for photosynthesis. Option 4, they protect chlorophyll A from photooxidation. So what is the role of xanthophylls and carotenoids in photosynthesis? Except ಇವೆಲ್ಲ ರೋಲ್ಸ್ಗಳಿವೆ. ರೋಲ್ಸ್ಗಳನ್ನ ಹೊರತುಪಡಿಸಿ ಯಾವ ರೋಲ್ ಅನ್ನ xanthophylls ಮತ್ತೆ carotenoids ಪ್ಲೇ ಮಾಡೋದಿಲ್ಲ ಅಂತ. Yes, YouTube students, what is your response on this? Yes, come on. Okay, see here. The answer is option B. Their peak position is in blue and red regions of Vibgyor. So blue ಮತ್ತೆ ರೆಡ್ ರೀಜನ್ಸ್ ಅಲ್ಲಿ ಇರೋದಿಲ್ಲ ಯಾವುದು xanthophyll ಮತ್ತೆ ಕೆರಾಟಿನಾಯ್ಡ್ಸ್ ಅದು ಕ್ಲೋರೋಫಿಲ್ ಇರುತ್ತೆ. ಸೊ ಆಪ್ಷನ್ ಬಿ ಇಸ್ ಅ ರೈಟ್ ಆನ್ಸರ್. ಆಪ್ಷನ್ ಬಿ ಇಸ್ ಅ ರೈಟ್ ಆನ್ಸರ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. ಕಂಪೇರ್ಡ್ ಟು ರೆಟಿನಾಲ್, chlorophyll can be described as a pigment that has a. ಸೊ ರೆಟಿನಾಲ್ ಅನ್ನೋ ಒಂದು ಪಿಗ್ಮೆಂಟ್ ಇದೆ. ಸೊ ಅದಕ್ಕೆ ಕ್ಲೋರೋಫಿಲ್ ಅನ್ನ ಹೋಲಿಸಿದ್ರೆ chlorophyll can be described as a pigment that has a narrow absorption range but high efficiency, narrow absorption range but low efficiency, wide absorption range but high efficiency, wide absorption rate but low efficiency. Yes, tell me. Compared to retinal, chlorophyll can be described as a pigment that has a. Yes, anyone? YouTube students, what is your answer on this? Okay, let us check the answer here. So retinal ಮತ್ತೆ chlorophyll ಅನ್ನ ನೋಡಿದಾಗ ಆಪ್ಷನ್ ಎ ನ್ಯಾರೋ ಅಬ್ಸಾರ್ಪ್ಷನ್ ರೇಂಜ್ ಬಟ್ ಹೈ ಎಫಿಷಿಯನ್ಸಿ. ಯಾರಿಗೆ? ಕ್ಲೋರೋಫಿಲ್ಗೆ ನ್ಯಾರೋ ಅಬ್ಸಾರ್ಪ್ಷನ್ ರೇಂಜ್ ಇರುತ್ತೆ ಬಟ್ ಅದರ ಎಫಿಷಿಯನ್ಸಿ ಏನಿದೆ ಜಾಸ್ತಿ ಇದೆ ಕಂಪೇರ್ಡ್ ಟು ರೆಟಿನಾಲ್. ಸೊ ಆಪ್ಷನ್ 1 is a right answer. Next question. The half-leaf experiment showed that dash is important for photosynthesis. So we have various factors which are important for photosynthesis, right? Carbon dioxide is necessary. Then what we can say, sunlight is necessary. Chlorophyll is necessary. Water is necessary, right? Chlorophyll, water. So we have various experiments which proves carbon dioxide is necessary, sunlight is necessary, chlorophyll is necessary, and water is necessary. Same way, half-life experiment showed that which is important factor for the photosynthesis: oxygen, carbon dioxide, sunlight, chlorophyll. Yes, what is your answer on this? The half-leaf experiment showed that, come on. Yes, very easy question, right? So we learned half-life experiment while learning the theory of the photosynthesis, ಅಲ್ವಾ? ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಹಾಫ್ ಲೈಫ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅಂತ ಬರುತ್ತೆ. ಇದನ್ನ ಥಿಯರಿ ಕ್ಲಾಸಸ್ ಅಲ್ಲಿ ನಾವು ಕಲ್ತಿದ್ದೇವೆ ಅಲ್ವಾ? Okay, let us proceed to the next question. The answer is here option B, that is carbon dioxide. Option B, carbon dioxide is necessary ಅನ್ನೋದು half leaf experiment show ಮಾಡುತ್ತೆ. Next question. A compound was used in the half leaf experiment to absorb carbon dioxide. This compound is ಯಾವ compound ಅನ್ನ ಯೂಸ್ ಮಾಡ್ತೀವಿ half leaf experiment ಅಲ್ಲಿ to absorb the carbon dioxide? Potassium permanganate, what we can say, hydrochloric acid, sodium hydroxide, potassium hydroxide. Yes. Yes, a compound was used in the half leaf experiment to absorb carbon dioxide. Which compound it is? Yes, YouTube students. So the right answer is option D, that is potassium hydroxide. It absorbs the what we can say carbon dioxide. Next question. Chlorophyll B is the major pigment associated with photosynthesis. So we have various pigments, chlorophyll A, B, D, E, right? So ಅದರಲ್ಲಿ chlorophyll B ಇದು major pigment ಆಗಿದೆ photosynthesis ಅಲ್ಲಿ ಅಂತ ಹೇಳ್ತಿದ್ದಾರೆ. Whether this statement is true or false? Is it true or false? Yes, any answers? Let us check. So the right answer is option A. It is true. Chlorophyll B is a major pigment. Next question. Which scientist showed that only the green part of the plants will release oxygen? ಯಾವ scientist green part of plant oxygen ಅನ್ನ release ಮಾಡತ್ತೆ ಅಂತ ಹೇಳಿದ್ರು? Option A, Jan Ingenhouse. Option B, Joseph Priestley. Option C, Van Neal. Option D, T.W. Engelmann. So which scientist showed that only the green part of the plants will release oxygen? Yes. Tell me. Yeah. Which scientist showed that only the green part of plants will release oxygen? So when you say then only we can solve more questions, Alva. So the right answer for this question is option A, Jan Ingenhouse. Jan Ingenhouse was the person who said only the green parts of plants will release oxygen. Next question. Who found the presence and properties of glucose in green plants? Green plants ಅಲ್ಲಿ ಗ್ಲುಕೋಸ್ ಇದೆ. ಅದರ ಪ್ರಾಪರ್ಟೀಸ್ ಅನ್ನ ಯಾರು ಕಂಡುಹಿಡಿದ್ರು? T.W. Engelmann, C. Van Leeuwen, Julius von Sachs, Jan Ingenhouse. Who found the presence of? Very easy questions, theoretical questions ಇದೆ ಈಗ. 10th question ಆದ್ಮೇಲೆ we will get the tough questions. So ಇವು theoretical questions ಇವೆ. Theory ಆಗಿದೆ ಇದು. We already completed this in theory classes. So in college also it will be finished, right? So do answer the question. Who found the presence and properties of glucose in green plants? Engelmann, Van Neal, Julius von Sachs, Jan Ingenhouse. So the right answer for this question is option C, Julius von Sachs. Julius von Sachs. Next question. The first action spectrum based on photosynthesis was given by. So action spectrum ಬಗ್ಗೆ ನಾವು ಅವಾಗಲೇ ನೋಡಿದ್ವಿ. Just a minute. Here we have regarding action spectrum. See here. ಸೊ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಮತ್ತೆ ಆಕ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಅಂತ ಬರುತ್ತೆ. ಸೊ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಅಲ್ಲಿ ಏನಿದೆ, ಇಟ್ ಈಸ್ ದ ಕರ್ವ್ ದಟ್ ಶೋಸ್ ದ ಅಮೌಂಟ್ ಆಫ್ ಡಿಫ್ರೆಂಟ್ ವೇವ್ ಲೆಂತ್ ಆಫ್ ಲೈಟ್ ಅಬ್ಸಾರ್ಬ್ಡ್ ಬೈ ಅ ಸಬ್ಸ್ಟೆನ್ಸ್. ಸೊ ಒಂದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಯಾವ್ಯಾವ ವೇವ್ ಲೆಂತ್ ಆಫ್ ಲೈಟ್ ಅನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡಿದೆ ಅದರ ಕರ್ವ್ ಅನ್ನ ತೋರಿಸುತ್ತೆ. ಯಾವುದು ಈ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್. ದ ಗ್ರಾಫ್ ಗಿವನ್ ಬಿಲೋ ಶೋಸ್ ದ ಎಬಿಲಿಟಿ ಆಫ್ ಕ್ಲೋರೋಫಿಲ್ ಎ ಟು ಅಬ್ಸಾರ್ಬ್ ಲೈಟ್ ಈಸ್ ಡಿಫ್ರೆಂಟ್ ಆಫ್ ಡಿಫ್ರೆಂಟ್ ವೇವ್ ಲೆಂತ್. Chlorophyll A shows the maximum absorption peak at 450 nanometer. ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಮ್ಯಾಕ್ಸಿಮಮ್ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಇದು ಕ್ಲೋರೋಫಿಲ್ ಎ ತೋರಿಸ್ತಾ ಇದೆ. ಸೊ 450 ನ್ಯಾನೋಮೀಟರ್ ಅಲ್ಲಿ ಕ್ಲೋರೋಫಿಲ್ ಎ ಜಾಸ್ತಿ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಮಾಡ್ಕೊಳತ್ತೆ. ಅದೇ 620 ಅಲ್ಲೂ ಕೂಡ ಇಲ್ಲಿ ನೋಡಿ, ಇದು ಕ್ಲೋರೋಫಿಲ್ ಎ ಅಲ್ವಾ, ಇದೆ ನೋಡಿ. ಸೊ ಇಲ್ಲಿ ಜಾಸ್ತಿ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಮಾಡ್ಕೊಳತ್ತೆ. ಹೌದಾ. ಕೆರೋಟಿನೈಟ್ಸ್ ನೋಡಿ, ಇಟ್ ಈಸ್ ಬಿಟ್ವೀನ್ 480 ಅಲ್ಲಿ ಹೈಯೆಸ್ಟ್ ಪೀಕ್ ತೋರಿಸ್ತಾ ಇದೆ. ಅದೇ ರೀತಿ ಕ್ಲೋರೋಫಿಲ್ ಬಿ ನೋಡಿ, ಇಟ್ ಈಸ್ ಶೋಯಿಂಗ್ ಅರೌಂಡ್ 450 ಆರ್ 470 ನ್ಯಾನೋಮೀಟರ್ ಅಲ್ಲಿ ಪೀಕ್ ತೋರಿಸ್ತಾ ಇದೆ. ರೈಟ್. ಸೊ ಅಬ್ಸಾರ್ಪ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಇಸ್ ಕಾನ್ಸ್ಟಿಟ್ಯೂಟೆಡ್ ಬೈ ವಯೋಲೆಟ್, ಗ್ರೀನ್, ಬ್ಲೂ, ಆರೆಂಜ್ ಮತ್ತೆ ರೆಡ್. ಈ ಲೈಟ್ಸ್ ಗಳಿಂದ ಅದು ಕಾನ್ಸ್ಟಿಟ್ಯೂಟ್ ಆಗಿರುತ್ತೆ. VBOR. ಎಮಿಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಇಸ್ ಕಾನ್ಸ್ಟಿಟ್ಯೂಟೆಡ್ ಬೈ ಯೆಲ್ಲೋ ಅಂಡ್ ಯೆಲ್ಲೋ ಗ್ರೀನ್ ಪಿಗ್ಮೆಂಟ್. ಎಮಿಷನ್ ಅಂದ್ರೆ ಯಾವ ಕಲರ್ ಎಮಿಟ್ ಆಗತ್ತೆ. ಆಕ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಅಂತ ಇದೆ. It is the curve that depicts the relative rates of photosynthesis at different wavelengths of light. ಈಗೇನು ಮಾಡಿ, I'm sorry. So it is the curve that depicts the relative rates of photosynthesis at different wavelengths of light. ಸೊ ಇಲ್ಲಿ ಏನಾಗತ್ತೆ ಅಂದ್ರೆ ರಿಲೇಟಿವ್ ರೇಟ್ ಅಂದ್ರೆ ಯಾವ ವೇವ್ ಲೆಂತ್ ಅಲ್ಲಿ ಎಷ್ಟು ಫೋಟೋಸಿಂಥೆಸಿಸ್, ಜಾಸ್ತಿ ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಯಾವ ವೇವ್ ಲೆಂತ್ ಅಲ್ಲಿ ಆಗಿದೆ, ಕಡಿಮೆ ಫೋಟೋಸಿಂಥೆಸಿಸ್ ಯಾವ ವೇವ್ ಲೆಂತ್ ಅಲ್ಲಿ ಆಗಿದೆ. ಸೊ ಅದನ್ನ ಡಿಪಿಕ್ಟ್ ಮಾಡುತ್ತೆ ಯಾವುದು ಈ ಆಕ್ಷನ್ ಸ್ಪೆಕ್ಟ್ರಮ್ ಅನ್ನೋದು. ಸೊ ಹಿಯರ್ ಬಿಲೋ ಶೋಸ್ ದ ವೇವ್ ಲೆಂತ್ ಅಟ್ ವಿಚ್ maximum photosynthesis occur at blue, violet and red wavelength in a plant. So see, blue, violet and red. ಹೌದಾ, ಇಲ್ಲೊಂದು ಪೀಕ್ ಇದೆ, ಇಲ್ಲೊಂದು ಪೀಕ್ ಇದೆ. Hence this concludes that chlorophyll A is the chief pigment. ಯಾಕೆ ಅಂದ್ರೆ ಅದು ಬ್ಲೂ, ವಯೋಲೆಟ್ ಮತ್ತೆ ರೆಡ್ ವೇವ್ ಲೆಂತ್ ಆಫ್ ಇದರನ್ನ ಜಾಸ್ತಿ ಫೋಟೋಸಿಂಥಸಿಸ್ ರೇಟ್ ಅನ್ನ ತೋರ್ಸತ್ತೆ ಅಂತ. ಫೈನ್. Okay. See, light absorbed is here. This is nanometer. This is the absorption what you can see. Fine. So let us proceed with the next question. The first action spectrum based on photosynthesis was given by anta ide. So the right answer is here option D, T.W. Engelmann. T.W. Engelmann. Right. Next question. Which of the following is the correct equation of photosynthesis? ಕೆಳಗಡೆ ಕೊಟ್ಟಿರೋದ್ರಲ್ಲಿ ಯಾವುದು photosynthesis ನ correct equation ಆಗಿದೆ? Look at the equations and let me know the answer. Okay, Anuradha answered it. Fine. So let us check the answer. So the right answer for this question is exactly option A. That is the right answer. The method by which leaf pigments of any green plants can be separated. So leaf pigments ಇರತ್ತೆ ಅಲ್ವಾ. ಸೊ ಪಿಗ್ಮೆಂಟ್ಸ್ ಅಲ್ಲಿ ಕ್ಲೋರೋಫಿಲ್ ಎ, ಕ್ಲೋರೋಫಿಲ್ ಬಿ, ಕ್ಸಾಂಥೋಫಿಲ್ಲು, ಸೊ ಇವೆಲ್ಲ ಇದೆ. ಸೊ ಎಲ್ಲ ಲೀಫ್ ಪಿಗ್ಮೆಂಟ್ಸ್ ಅನ್ನ ಯಾವ ಮೆಥಡ್ ಇಂದ ನಾವು ಸಪರೇಟ್ ಮಾಡಬಹುದು? ಕಂಡೆನ್ಸೇಷನ್, ಸೆಂಟ್ರಿಫ್ಯೂಗೇಷನ್, ಪೇಪರ್ ಕ್ರೊಮ್ಯಾಟೋಗ್ರಫಿ, ಡಿಸ್ಟಿಲೇಷನ್. Very easy question. The method by which leaf pigments of any green plants can be separated is called as. Option C. Exactly, paper chromatography. And Anuradha answered it correctly. Yes, YouTube students, that's right, option C. Next question. The following diagram represents Joseph Priestley experiment, Gregor Mendel experiment, Engelmann experiment, Linnaeus experiment. Whose experiment this diagram is representing? The following diagram represents. Yes. So when we study these notes, na, so here you can see. See, Priestley's experiment. Mouse dies in foul air. CD. Mouse survives in air purified by the mint plant. Right? So it is a Priestley experiment. So what he concluded by this experiment? Priestley hypothesized that the foul air produced by the burning of candles and a mouse respiration could be converted into pure air by plant. Okay. Yes, very good. So option A, Joseph Priestley ಅಂತ ಹೇಳ್ತಿದ್ದೀರಾ. Let us check the answer because I don't remember the options here. Exactly, Joseph Priestley, option A is the right answer. Okay, now. So here, during which year Joseph Priestley discovered oxygen? Joseph Priestley ಅವರು ಆಕ್ಸಿಜನ್ ಅನ್ನ ಡಿಸ್ಕವರ್ ಮಾಡ್ತಾರೆ. ಯಾವ ಇಯರ್ ಅಲ್ಲಿ? 1774, 1770, 1884, 1880. Do refer your textbooks, do refer your reference books or notes and do answer the question. During which year Joseph Priestley discovered oxygen? See here. So by this experiment, what he concluded, burning of candles in the poisonous gas of the whole air release agate. That is why mouse died. So when the mint plant was kept, na, so that air was completely purified by the mint plant. So the mouse survived anta. So idrinda. Yes, tell me. Joseph Priestley, our experiment. He performed series of experiment in 1770. ಮತ್ತೇನನ್ನ ರಿಲೀವ್ ಮಾಡಿದ್ರು ಅವರು? Essentiality of air in the growth of green plant. Essentiality of air is nothing but essentiality of carbon dioxide and water. Then on the other hand, okay, ಇದು ಅವ್ರ experiment ಅನ್ನ explain ಮಾಡಿದ್ದಾರೆ. So let us check the answer here. The right answer is option A, 1774. Okay, next question. Photosynthesis is the basis of life on earth. Photosynthesis ಏನು? It is the base of life on earth. Because ಯಾಕೆ? It is the main source of all food on earth. It helps in the release of carbon dioxide into the atmosphere. It produces new offspring. It gives no energy to plant. Photosynthesis is the basis of life on earth because ಯಾಕೆ? Very good. So Anuradha answered it option A anta. Exactly the right answer. Other than carbon dioxide and light, carbon dioxide ಮತ್ತೆ light ಅನ್ನ ಬಿಟ್ಟು, which is used as the raw material for photosynthesis. Oxygen, carbon dioxide, water and manganese oxide. Other than carbon dioxide and light, which is used as the raw material for photosynthesis. Very easy question. When you remember the reaction of photosynthesis, na, then you can understand it. Recall the reaction of photosynthesis. So which are the raw materials or the reactants we need? Yes, correct. Option C. Yeah. So exactly it is water. Oxygen is a product, right? Carbon dioxide is one of the reactant. It is already given. So the remaining is water. Next question. The first acceptor of electron from an excited chlorophyll molecule of photosystem 2. So photosystem 2 ಬರುತ್ತೆ ಅಲ್ವಾ photosystem 2 ಅಲ್ಲಿ ಫಸ್ಟ್ ಎಲೆಕ್ಟ್ರಾನ್ ಎಕ್ಸೆಪ್ಟರ್ ಯಾವುದು ಎಲೆಕ್ಟ್ರಾನ್ಸ್ ಎಕ್ಸೈಟ್ ಆಗ್ತವೆ by absorbing the photons. Those electrons are accepted by the electron acceptor. So which is that electron acceptor in PS2? Quinone, cytochrome, iron sulfur protein, peridoxin. Peridoxin. So when you see a photo system 1 and 2, let me check whether we have a detailed notes on it. Okay, answer the question. Okay, B, cytochrome anta. So the answer is here PS2 ಅಲ್ಲಿ quinone will be a electron acceptor. Quinone. They asked PS2. Okay. So option A is the right answer. Next question. The primary acceptor of carbon dioxide in C3 plants. C3 plants ಅಲ್ಲಿ ಮೊದಲಿಗೆ primary acceptor of carbon dioxide ಯಾರು? RUBP, PGA, PEP, malic acid. Yes. Yes, tell me. Oh, just a minute. Okay, Anuradha answered it. Just a minute. So here you have C3 ಮತ್ತೆ C plants, C4 plants ಅಲ್ಲಿ ಇರೋ ಡಿಫ್ರೆನ್ಶಿಯೇಷನ್ ಅಥವಾ ಕಂಪ್ಯಾರಿಸನ್ ಇದೆ ಹೌದಾ ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಯಾ ಸೊ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಎಕ್ಸೆಪ್ಟರ್ ಯಾರಿರ್ತಾರೆ C3 ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಇಟ್ ಈಸ್ ರೈಬಿಲೋಸ್ 1,5 ಡೈಫಾಸ್ಫೇಟ್ ಇಟ್ ಈಸ್ ರೈಬಿಲೋಸ್ 1,5 ಡೈಫಾಸ್ಫೇಟ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಎಕ್ಸೆಪ್ಟರ್ C4 ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಫಾಸ್ಫೋಇನಾಲ್ ಪೈರುವೇಟ್ ಇರುತ್ತೆ ಸ್ಟೇಬಲ್ ಪ್ರಾಡಕ್ಟ್ ಯಾವುದು C3 ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಫಾಸ್ಫೋಗ್ಲಿಸರಿಕ್ ಆಸಿಡ್ PGA ಅದೇ C4 ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಸ್ಟೇಬಲ್ ಪ್ರಾಡಕ್ಟ್ ಯಾವುದು ಫಸ್ಟ್ ಸ್ಟೇಬಲ್ ಪ್ರಾಡಕ್ಟ್ ಆಕ್ಸಾಲೋ ಅಸಿಟೇಟ್ ಅಂತ types of chloroplast all cells participating in photosynthesis. The chloroplast of parenchymatous bundle sheath is different from mesophyll cells. Leaves have crans type of anatomy. The bundle sheath chloroplast lack grana. Mesophyll cells have normal chloroplast. ಇದು C4 plants ಅಲ್ಲಿ cycles ಅಲ್ಲಿ only reductive pentose phosphate cycle is formed. ಅಲ್ಲಿ both C4 dicarboxylic acid and reductive pentose phosphate form ಆಗುತ್ತೆ right so this is about C3 and C4 so Anuradha answered it YouTube students answered it option B okay so the right answer for this question is option A RUBP just now we discussed it alva ribulose diphosphate okay carbon dioxide acceptor in C3 plants next question choose the correct statement about C4 plants C4 plants ಬಗ್ಗೆ ಇರುವ correct statement ಅನ್ನ ನಾವು choose ಮಾಡಬೇಕು mesophyll cells possess rubisco neither mesophyll cells nor bundle sheath cells possess rubisco mesophyll cells have PEP case which convert carbon dioxide into OAA, oxaloacetic acid. Bundle sheath cells synthesize OAA, which is a 4-carbon compound. So, which is the correct statement about C4 plant? Just now we discussed C3 and C4, right? So, tell me now, what is your answer on this? D. Okay, option D. Okay. So, the right answer for this is option C. What is that? Mesophyll cells have PEPKs which convert carbon dioxide into oxaloacetic acid. Next question. Calvin cycle takes place in only C3 plants, only C4 plants, both C3 and C4 plants, neither C3 and C4 plants. Calvin cycle takes place in. Okay. Option A only C3 plants ಅಂತ. Option C both C3 ಮತ್ತೆ C4 ಅಲ್ಲಿ ಕ್ಯಾಲ್ವಿನ್ ಸೈಕಲ್ ಆಗತ್ತೆ. So regarding photosynthesis, again we are going to take one more session. So after that, we are going to complete the photosynthesis and we are going to start with the respiration in plants. Okay. So let me meet you all in the next session. Until that, take care. Have a nice day. Bye-bye.